everybody. And as Joe said, welcome to the webinar today, this morning, this afternoon, or this evening, depending on your time zone. And so, as Joe mentioned, what we are going to do today is focus on IntelliCyt's high-capacity flow screening platform and how that might be able to help you do better antibody screening and improve your productivity and find some interesting new antibodies to take through your development platform. So today I'm going to, here we go, this is the next screen. In today's webinar, what I'm going to do is do a couple of things. The first, what I'd like to do is talk about high-capacity flow and our platform, the HTFC screening system, and give you a background on that and how it might fit into your antibody screening efforts. And then what I want to do is talk about two specific applications that the platform can be used for in the world of hybridoma screening. One that looks at cell surface antigens, and the other looks at circulating antigens. And we'll talk a little bit more in detail about the technology and how it can be used for those two types of assays. And then we should have time at the end for some questions. And so there's a mechanism in this GoToWebinar product that allows you to ask questions at the end. So we'll try and make sure that we leave time at the end for that. So just a few slides about therapeutic antibody development. It really is a great place to work and to be doing interesting science these days. And there's a lot of reasons for that in terms of business and billions of dollars in sales of products. This is the fastest growing therapeutic modality that's out there. Biologics is really growing fast. And there are currently, according to the latest reports, there are 28 approved antibodies out there now for therapeutic indications, and there are a ton of antibodies in clinical development. At last count, there's around 350 antibodies that are going through some stage of clinical development at this point. And as many of you, I'm sure, realize, therapeutic antibodies are highly specific, and so there's not that the chance for off-target toxicity is relatively low, and what that translates into is a high rate of clinical success. So I have this graph here on this slide that charts the transition probability through the different phases of clinical development of antibodies, and you can see it's broken down into cancer antibodies and some immunological targets, but basically the bottom line to take away from this chart is that there's a high rate of clinical success going from phase 1 to 2, from 2 to 3, and from 3 to approval. So that's one of the main reasons why therapeutic antibodies are really growing as a modality these days. But one of the best things for all of us is that really these antibodies, these therapeutics, are really starting to make a difference in patients' lives, and that's a very exciting place to be. And all I've done here is listed just a handful of some of the approved antibodies that have been developed for various indications, and anywhere from cancer to MS to asthma and RA. There's a lot more antibodies out there, and it's a very exciting place to be because really helping patients and improving people's lives. So, as I'm sure all of you know, antibodies, it's difficult to get in a therapeutic setting. It's difficult to get immunoglobulins and antibodies inside of cells. So what that means is that really there are two classes of targets that are tractable by antibodies, monoclonal antibody therapies. And they basically have to be outside of the cell. So cell surface antigens are tractable, receptors, ion channels, things like that are popular targets for antibody therapies. And of course, the other class is going to be circulating antigens like cytokines and hormones, growth factors, things like that. These numbers in parentheses next to receptors and circulating proteins and viruses just indicate some of the number of, according to reports, candidates in late-stage clinical trials now for each of those classes. But anyway, the space is really quite focused on these two types of targets. And what that means is that the competition out there amongst all of you guys doing therapeutic antibody development is pretty intense. And this is a slide that I put together just kind of gathering the different antibodies and their targets and their indications. And what it really highlights is there's a lot of people going after the same targets. There's 3 targets listed here. One is TNF-alpha for rheumatoid arthritis, and there are 4 approved antibodies for that against that target at the moment. CD20 down there at the bottom, I believe the number is 10 or 12 antibodies there that are either approved or in development for cancer and rheumatoid arthritis, and then the insulin-like growth factor receptor. There's quite a hot target these days. A lot of antibodies in development for that. So there's a relatively small number of targets, and there's a lot of people going after those same targets. In addition, there's a lot of really neat antibody technologies out there that are in the discovery area, and so of course it all started with mouse hybridomas and making hybridomas out of mice, and then humanization and transgenic mice affinity maturation, phage display. There's just a lot of antibody discovery technologies that are being developed now, and some very interesting ways to try and find new targets and find new ways of developing antibodies to get at those targets. But what all these have in common is really they all are going to need some powerful screening tools to be able to sift through your antibody libraries to find those antibodies and engineer those antibodies that are going to be able to have the biological effect in the clinic. And so really what we're really needing here is something beyond ELISA. ELISA, of course, is the gold standard for antibody screening, and I think we're getting to the point now where we're really looking for new tools, powerful tools that can help us take advantage of some of these discovery technologies. So we've discussed a lot about the ideal antibody screening platform that you guys could use, and what are the kinds of things that could really take advantage of some of those technologies that were on the previous slide. And I've constructed a list here, and we'll go through some of these in a little more detail later. But let's see if I can get my pointer working here. So an ideal screening platform in the antibody therapeutics world would really be able to get at both of those types of targets that I mentioned earlier: cell surface targets and circulating targets. So the ideal platform to be in your lab on the bench working every day is one that can go back and forth between looking at cell surface antigens and circulating targets. For the cell-based assays, you'd want also to be, of course, to be able to look at suspension cells, so that if you're looking at cells of the immune system, or if you're looking at cancer cells, you want to be looking at both types of cell formats. So an ideal situation would be a platform that can do both. You would like to be able to look at single cell or particle measurements, and also be able to do multiplex to be able to put out multiple readouts per well. And that allows you to do phenotypic screening, which is a very powerful technique for really understanding what's happening with your library as it's going through your screen. With any screening technology, you'd like to be able to reduce the number of washes that you need to do to increase your signal to background. And so, of course, the ideal situation would be to have no wash capabilities. And of course, we're going to want to have, for a screening tool, we want to have it high speed, high throughput, amenable to setting up with plate handling robots and liquid handling robots, and of course easy to use so that it's you can walk up to it and run it.
So Tom, I wanted to ask you, this is Joe, this easy to use, right? We talk about it a lot, and I just wanted to touch on the fact that it has numerous components, right? There's the plate preparation, getting a hold of the reagents, having them available and locked down, essentially pre-developed, but also the ability to go through and be able to develop an assay very quickly in a reproducible way and have it be able to be a screening tool. So easy to use has a lot of value. So
that's correct. And one thing that we are one of our missions here at IntelliCyt is we desire, and our goal is to be a complete solution provider. So we are, as you'll see, our product line includes platforms for screening, but it also includes software and data analysis software and screening software, but also reagent kits and consumables that help you set up an assay and run it from a very easy to use standpoint. Okay, so this is a diagram of our picture of our platform product called the HTFC screening system, and we're going to talk a little bit about this platform, this screening platform, and how it can be used to help your antibody discovery efforts. So just walk through. I want to spend just a minute walking through the components of the system to give you guys a flavor for how this thing works. As I mentioned in the title slides and earlier on, this is what we call a high-capacity flow system. So this is a high-throughput system that uses flow cytometry as its detection module. And that imparts some very interesting and powerful capabilities into a screening system. So the HTFC detection is basically a flow cytometry detection module. It has 2 lasers and 6 channels of data. It's very stable in terms of instrument to instrument, but also from day to day performance. So people can, different people in the lab can walk up and use this and get the same data that somebody else is generating. It has 6 decades of dynamic range, which means that you can look at in the world of hybridoma screening, you could look at low binders and you can see high binders in the same well even of your screening plate. So it's got a nice large dynamic range. It's fast. It uses a proprietary sampling technology that we have here at IntelliCyt, which allows you to run a 96-well plate in as fast as 3 minutes and a 384-well plate in as fast as 12 minutes. With everything optimized, that's very fast sampling speed, and that makes it for a nice throughput for hybridoma screening. And then on the back end, what we have is a screening and analysis software that we call Foresight software, which allows you to do a number of things. It goes very quickly from data that's fairly complicated underneath the hood to present hits and trends in heat map format, which we'll show you a little bit later. But it's able to run multiplex data and do analysis very easily on that, and it allows you to find hits very quickly. And it's also very scalable, which we won't talk about today, but this basically allows you to manage your data across your entire organization. And finally, there is hooks in this software that allow you to plug into plate handling robots and other types of robots if you are interested in automating your process. So a little bit about the throughput of the system. I mentioned the timing in terms of scanning a 96 or a 384-well plate. And these are just some numbers that we sort of went through and did the math. So if you assume, for example, 15 minutes for a 384-well plate, and start crunching through the number of wells per hour and the number of plates per today, you can see very quickly that that you can, the system has the potential of running, nearly 500 plates a week and over 180,000 wells per week, and that is allows you to do quite a number of hybridoma screens, which just improves your chance of success. So you're able to really kind of by screening more wells, you're able to take advantage of what screening is, which is to find the needle in the haystack, and that improves your chances of success. So one thing about the HTFC screening system that's different than a lot of other systems, though, is this ability to read multiple readouts in every well. And we're going to talk about this a bit on the next few slides. But if you take these numbers of 180,000 per week and then you start looking at the ability to look at 2 outputs per well, or 4 outputs per well, or 6 outputs per well, you're starting to get into the million data points per week, and that becomes an extremely powerful screening platform when you take all that into account. Okay, so what I'd like to do now is give you two examples, or talk about 2 examples that pertain to the 2 types of antibody targets that are available or that are tractable by these therapeutic antibodies. And so first I'm going to talk about cell surface target antigens, and then a little bit later I'm going to talk about how we can screen for antibodies against circulating target antigens. But let's talk about cell-based assays first. So one of the things that we can that's clear in the last few years is that it's becoming very important. If you are screening for antibodies against cell surface antigens, it's important to set up a screening platform and a screening assay that tests the binding of your library to the antigens in their natural conformation. And so this is sort of depicted here, that if you can set up a cell-based assay, your target antigen is going to be in its natural conformation, which is opposed to a standard plate-based ELISA assay, where the antigen has been purified and pulled out of its natural conformation, because what that potentially does is disrupts your conformational epitopes, and there are a lot of interesting antibodies that can be developed against conformational epitopes that you might not see in a typical plate-based ELISA screen. And I mean, I can tell you when I was back at Tanox screening for antibodies, we did some side-by-side analyses using cell-based assays versus plate-based ELISAs, and we really we found out that we were missing at least 30% of antibodies that were binding in a cell-based assay were not even showing up in an ELISA assay. So we were potentially missing some very interesting antibodies that could be used downstream in the clinic. So one of the things that I want to talk about now, just for a second, is we have a technology here that we call the cell encoder technology, and our hybridoma screening assay is really kind of built around this. And I'm going to walk you through this. It's a fairly simple technology, but I think it's important to walk through and just so we're all on the same page here. So this cartoon shows 2 cells, cell 1 and cell 2, and you can imagine that cell 2 has a target antigen on its surface, and cell 1 does not. So what we can do with the encoder technology is basically code or color one of the cell types and not the other, and now we can use that downstream to multiplex this assay. So in this example, you would take our encoder dye and add it to cell 2, but not to cell 1. Wash away the unbound dye, and what you will end up with are cells. The cell 2 is green and cell 1 is not green. This is green fluorescence. And what you can do then is combine these two cell types, one is green and one is not, in the same wells of your screening plates. And now what you've done is you've set it up so that you can do multiple assays per well. So what you can do then is once you have the cells combined in your wells, add your test samples, for example, a hybridoma library with some detection reagents, fluorescent dye, and I'm sorry, I'm having a bit of a technical here. And then what you can do is add a detection reagent and look at, for each cell in every well, you can look at the biology or the chemistry that is representative of that. And so what you can really do is, okay, hang on one sec, this, what you can do is look at separate readouts for every well. And so what that really allows you to do is run multiple assays in every well. So now in this example, instead of running a separate screen for a binding event for the cell 2, you can also in the same well simultaneously look at cell 1 as well and look at binding events to that. So it's in this very simple example, you're really doubling your productivity and your throughput. Now what I want to do is take you through a specific example of how that can be used. And this is basically a similar diagram to what you saw before. So we've got target cells. These cells are not being labeled with our encoder dye. And we have control cells that are being labeled. We mix them together. We add our hybridoma supernatants and a detection reagent that's going to detect in a different channel than this green encoder dye. And what we'll be looking for are antibodies that light up against the target cells, but not to the control cells. So if you look down here at the lower panel, this is a little bit about what the data looks like. So if you can, what we're actually doing here is as an example, we're looking for anti-CD45 antibodies. And so you see our encoder dye here is represented, the intensity of the encoder dye is represented here on the x-axis. So what we see here is we see a nice distinct population of our target cells here, and the control cells, the CD45 negative cells, are shown in the green population here. And once we then put that through the HTFC screening system, adding our hybridoma supernatants, adding our detection reagents, we can then, for each plate of our screen, we can generate a separate heat map. So this, what you see on the right here is a heat map of the same plate. The top one is for the control cells, for the CD45 negative cells, and the bottom one is for the target cells. And what you can easily do here then is you can look to see where your hits are and where your specific hits are and your cross-reactive hits. So if you look at the C11 here, you'll see a specific hit that lights up on the target cell plate but not on the control cell plate. And conversely, you can see a cross-reactive hit that's lighting up here in H10 that's lighting up both on your control cells and on your target cells. So you might want to take these specific hits and pursue them further. So it's a very quick way of building in internal controls into each well of your plate, easily identify cross-reactive antibodies. And one thing I didn't mention about this is that we have been able to develop this as a no-wash assay, and it actually works very well. And so we've built and we have on the market now a multi-site cell-based hybridoma screening kit, which includes our encoder dye and detection antibodies and protocols, and of course a great support team that will help you you build your assays that can do this multiplex screening on the HTFC screening platform.
So, Tom, this is Joe. This multiplexing thing looks very, very exciting because it allows you to get rid of the pipetting error that you might have by trying to do 2 different ELISAs at 2 different times and 2 different incubators. So it really kind of lowers the innate variability in the assay. Can this be done with more than like 2 cell types at a time?
Yes, and we're gonna talk about that in a second. Actually, one of the other things to mention too is that with having control cells in the same well of every plate, you have an internal reference for your binding capacity against your target cells, and that's kind of shown here in the next slide. So this is a little bit of data to demonstrate the range of this assay. This was data generated from a no-wash, the no-wash screening assay that we talked about in the previous slide with using looking for an anti-CD45 antibody. And what we can see here is with this no-wash assay, first of all, you see this hook effect at the top, which is well characterized when you do no-wash assays and are looking at very high concentrations of antibodies in your supernates. But what this shows is that the range here is between about 1 and 50 micrograms per mil where you can see a nice differential staining binding effect between the negative cells here in red, or the control cells in red, versus the target cells, the binding to the target cells in blue. And that's, in our experience, we usually have between 1 and 10 micrograms per mil of antibodies in our hybridoma supernate. So this is right within range of those concentrations. So this is a very powerful way to do hybridoma screening. You're including controls in every well of the plate, which just increases your confidence. It increases your productivity, and gives you a better chance at success. So as Joe mentioned, you can take this encoder technology and really supercharge it by doing more than 2 cells per well. So I wanted to show you an example of that. And before I do that, just to sort of throw out a potential problem. This is a problem that we ran into over and over again, and that is you make a nice antibody that's got high affinity to a human target antigen, because that's the ultimate goal is to take these antibodies into people. But what can happen from time to time is that those antibodies are specific to the human target antigen, but they don't cross-react with the animal versions of the antigen, and that prevents you then from using that candidate antibody in downstream models of efficacy and tox. And so one of our customers actually is using our HTFC screening system to get at that problem by trying to identify and build into their screens candidates, antibody candidates that actually cross-react both with the human target antigen and target antigens from other species that can be used in downstream models. So just a quick overview of how that might work. This is again using the encoder technology, but here we're going to use 5 different cell lines. Each cell line is expressing a receptor from a different, the same receptor but from a different animal species. And so we use the encoder technology to color code these cell lines. We can easily distinguish them using the HTFC screening system, and then we put them in the screening place, and then we can detect the binding of our candidate antibodies to each one of those cell lines independently, but more importantly, simultaneously. So we're basically here doing 5 different assays in every well. Okay, what I want to do now is switch gears a little bit and talk about how we can use high-capacity flow and the HTFC screening system to screen for antibodies against the other class of tractable target antigens, and those are antigens that are circulating in the blood or the lymph, things like cytokines, growth factors. And the way we're approaching this is with bead-based ELISAs. And using the HTFC screening system, as you'll see, this is actually a very powerful way of taking a standard ELISA and making it way more powerful than a standard plate-based ELISA. And so just one thing I wanted to mention is whenever you see these three words, bead-based ELISA, what often comes to people's minds are some of the technologies that are out there that are very powerful, such as Luminex and the cytometric bead arrays that are used. And what I wanted to just kind of get the point across is what we're talking about here is something very different. What I'm going to show you is a way that you can take what you're doing now with your standard plate-based ELISA and translate that into a bead-based ELISA and take advantage of all of the things that you can do with that. So let's just take a very basic look at a plate-based ELISA. So when you're doing hybridoma screening, usually what you're doing is you're immobilizing your antigen down on the bottom of your ELISA plate, and then coming in with your hybridoma soups or your antibody library and detecting that with some sort of detection reagent. So what we're going to do with the bead-based ELISA is really the same thing, but rather than immobilizing your circulating antigen on the bottom of a plate, we're just going to use a bead to immobilize that antigen, and then we're going to come in and put those beads in the well of a screening plate, and we're going to come in and target that with our hybridoma soups and a detection reagent, very similar to the plate-based ELISA. But what this allows us to do now is you can imagine that you can only put one antigen down on each well of the plate, and usually it's the whole plate at a time for a plate-based ELISA. Whereas with a bead-based ELISA, you can very easily see how you can multiplex and look at two different antigens on two different beads in the same well of every plate. And what we'll show you is you can even expand this into more than two antigens.
So Tom, I'm looking at this, and what strikes me is the surface area difference, right? So it looks to me like you would be able to use less antigen, potentially, by binding them to the beads.
That's correct. One of the things that we have that's embedded or inherent in the HTFC screening system is that we're able to make our volumes very low, and so we can reduce the volumes of the beads of the solution that the beads are in very low and concentrate them, so we can get the same sort of surface area in these spherical beads that we can get on the bottom of a flat plate. And that allows you to do some interesting things in terms of increasing the capacity of binding and things like that. So it's a very good point. Just wanted to take you through a very quick example, a very simple example here of an assay where we were looking at antibodies that cross-react, similar to what we talked about before, but looking at an antibody that can cross-react with both human and mouse interleukin-5. So we took two different beads. Each bead has been impregnated with a different intensity or different concentration of a green dye. So that's why this human IL-5 has been coated onto a dark green bead, and the mouse IL-5 has been coated onto a light green bead, fluorescently speaking. And then what we can look at to see if antibodies bind to each one of these populations using the HTFC screening system. And some of the data that's shown on the next slide is just that this is an antibody that is binding to the beads that have been coated with mouse IL-5, and then titration of that same antibody that is binding to beads that have been coated with human IL-5, and then our control beads are not showing any binding at all. And so this again allows you to do some very interesting things in your hybridoma screens, as we talked about before. Similar to the cell-based screens, you can build negative controls into control antigens into every well. You can easily identify cross-reactive antibodies, and we have we've also had some data to show that this can be done also in a no-wash format using the HTFC screening system. This is a little bit of data, example data showing, again, the range of antibody, of detecting antibodies in supernatants. So these are just looking at two different antigens. One is an FC tagged, a ligand for the eprin A2 receptor, and the other is IL-17, which is a very popular cytokine target these days. And you can see that it depends on the antigen and the antibody that you're using, but the range is basically pretty broad here. So like in this case, the background signal is in blue, and the actual binding to the target leads is in red, and you can see that there's a nice signal to noise, and we're looking at anywhere from 24 nanograms up to 25 micrograms per mil range for this FC tag antigen, and anywhere between 200 and 25 nanograms microgram, 2.2 and 25 micrograms per mil for this IL-17 model that we use. These are, again, within this design to show you that, using in the range of antibodies that you have in your hybridoma supernates, you can take these, add them directly to these bead-based assays, and get nice signal-to-noise windows. As with the cell-based encoder technology, we were able to use our encoder technology to expand and do some more complicated bead-based ELISAs. For example, if you're looking for cross-reactive antibodies against a number of different antigens, this is expanding on that notion where we've got 7 different beads, each one incorporated with a different level of green dye, and then looking at the ability of an antibody to bind to each one of those antigens. Very easy to distinguish the separate populations. This is a little snapshot from our software. And then what we do in the Foresight software is we report heat maps. And so these are heat maps, again, from a single plate, but looking at each one of these different antigens, binding of the antibody to each one of these different antigens, so antigen 1 through 7. And you can see some specific antibodies that bind only to antigen 1, and you can see some that cross-react with antigen 3 and 4 and 5 and so on. So very powerful and very quickly expands your capabilities and your capacity for doing screening assays. And one thing I just wanted to sort of touch on is a little bit of an ROI analysis. So this is a table that's comparing the time and cost for doing a plate-based ELISA versus a high-capacity flow bead-based ELISA using the HTFC screening system. And these are some estimated costs on the reagents and plates and things like that for doing these two types of assays. So we would estimate about 100 minutes to do the whole thing. This is staining and washing and everything using a no-wash assay that we've developed here for this bead-based ELISA versus plate-based ELISA, which you would have to wash. So we're looking at about 100 minutes for the bead-based ELISA versus 75 for a plate-based ELISA, about the same cost for each. And if you just kind of conceptually think about the number of plates to do this 10-plate screen with a single antigen, obviously it's going to be 10 and 10. Where the real interesting comparison comes into play is if you're starting to multiplex. So if we look at 2 antigens on the HCF bead-based ELISA, it's still going to be the same time. It's going to be a little bit of increase in cost in beads, but you're still only going to be able to need to run 10 plates for this two antigen screen. Whereas with the plate-based ELISA, you basically have to run a plate for each antigen, or 10 plates for each antigen. So basically doubling your time and your cost, and then you can start to do the math a little bit. You probably see where I'm going here, where if you start to do 4 antigens, 6 antigens, 8 antigens, you can start to really see the time and cost savings add up with using the high-capacity flow bead-based ELISA. So just to sort of wrap up here, we're sort of starting to get out of time here, so I wanted to wrap up a little bit and revisit this checklist that we presented in the beginning. And so if you're looking at an ideal antibody screening platform, you want something that's going to be able to do cell-based screening so that you can screen against cell surface target antigens in both suspension and adherent cell formats. The same platform would be ideal to be able to look at binding assays, so basically an ELISA replacement for circulating target antigens. You would like to do single cell, single particle measurements so that you can look and see if your antibodies are binding to a separate population or not, but also helps you or allows you to do multiplexing where you can look at predefined different populations using for example, the encoder technology that I mentioned before. Well, again, with any screening system, if you can do no-wash assays, you're ahead of the game because it really reduces your cost and your time in terms of processing your plates. You would need to have high throughput, automation-friendly, and easy to use. And we are proposing that the HTFC screening system meets all these criteria, and so would make a great addition to any hybridoma or any antibody screening platform environment that you guys are using. And so just to let you know, just wanted to finish here to let you know that we do have a large customer base for the HTFC screening system, and it spans across large pharma, mid-pharma, small biotech. And these are some of the companies. There's a whole list of them, but there's a lot of people who are starting to use high-capacity flow for antibody screening because of all of the attributes and the qualities and the features that I mentioned before. Makes it an ideal way to do hybridoma screening and other types of antibody screening. So I'll wrap it up there. Thank you very much for attending, taking the time out of your busy schedules to spend some time with us.